Let me go ahead and introduce you then. Indika Rajapakse obtained a Bachelor of Science in Electrical Engineering and a PhD in Applied Mathematics and pursued postdoctoral work at Fred Hutch with Dr. Mark Gradine. Indika is currently an Associate Professor of Computational Medicine and Bioinformatics at the Medical School, an Associate Professor of Mathematics at the College of Literature, Science, and the Arts, and an Associate Professor of Biomedical Engineering in the College of Engineering at the University of Michigan, Ann Arbor. He's also a member of the Smale Institute and is the Chief Scientific Officer and co-founder of Reprogramming LLC. So with that, Indika, I'm really excited to hear your talk.
Oh, thank you so much. Thank you so much, Alex, for inviting me. And I will tell you more about what I'm doing, and maybe I might divert a little bit my topic in the later, the reprogramming, but please disturb me anytime during my talk. And first, so my lab is funded by Defense Advanced Research Agency project and the Air Force Air Force Office of Scientific Research. And so I can do a little bit so I always use to connect with beautiful biology with beautiful math and engineering, and also there's an institute called Smale Institute and a company I helped to start called Reprogram. And first I want to start that I came to really who I am is very unexpectedly. And all of my things I'm telling you today responsible from Dr. Grudin. I learned all of my biology from Dr. Grudin, Mark Grudin. And I'm so grateful for him. And Stephen Smale, he's a mathematician at UBerkeley. So I closely work with Steve for many years now. And then my main inspiration also my wife, Lindsay. And Lindsay did her PhD in molecular and cell biology program at University of Washington. So she's one of the reasons I switched to biology. And so before I start, I want to start with this. So I'm originally from Sri Lanka, and it's a small island just below India. And the chancellor of my university back in Sri Lanka was Arthur C. Clarke. And I never knew when I was back in Sri Lanka that how he was just a guy, he was the chancellor, but I think he guided us to think differently. But I realized after move to US that he's well known here. But I because of his book 2001 Space Odyssey, and I don't know how many of you know that there are 3 laws by Arthur C. Clarke, and I really like the 3rd law. So that is any sufficiently advanced technology is indistinguishable from magic. So for me, that connecting beautiful biology with so much beautiful mathematics and engineering, that's like magic at the end. So I will share with you a few things I have been doing last few years. I'm constantly learning, by the way. And I realize more and more I don't know so much. So the things I've done, I will share with you. And please disturb me anytime, please. And so this is my plan. I will give you a basic introduction to cell reprogramming. Specifically a technique I developed, and not myself, some few students of mine and myself, we develop a technique called data-guided control. And then number 2, I will tell you my recent work last 1.5 years on how to accelerate skin regeneration. And thirdly, I will talk to you a new technology called POSI. That's a beautiful technique to see inside the nucleus. And 4th, I will introduce to you a new thing called Turing system. These are all connected to cell reprogramming, So the first, reprogramming. This is like a simple for me, give you a brief summary. All cells of the body have the same set of genes, roughly. And adult cells of one kind can be reprogrammed either through embryonic stem cells or directly to cell of an unrelated kind. Right? And I will tell you this more detail. The replacement of some kind of aged or non-functional cells in human is real prospect in the future. An application of reprogramming. One is cell replacement therapy. And one thing I am very much into is how to create a hematopoietic stem cell from a skin cell directly. Number 2, wound healing. Number 3 is a little bit science fiction to me still. Printed a 3D tissue from skin fibroblasts, just purely from skin fibroblasts. So that is what at the end, I will tell you a bit about like science fiction, but at the end. So roughly the humans have 10 to the 30 number of cells in the body. Human heart has 5, 10 to the 11. And if you have a problem in the heart, then that reduced to 10 to the 11. And retinal pigmental epithelium of the eye have roughly 50,000 cells, small number of cells. Why I'm saying is that like retinal pigmented epithelium of the eye, someday you can replace the problematic cell from reprogramming. It's a small number of cells. And in fact, the heart too, that people have been working in a group at UCSF, that I think what they're claiming is that they can, in the heart is built with 2 types of cell types, cardiac fibroblasts and cardiac myocytes. I think my understanding is Dr. Chuck Murray might be here in the audience. So they are doing that. Can you directly reprogram cardiac fibroblasts to become a cardiac myocytes? I think these are all possible in my mind directly. So here I mentioned one thing. Can you create a hematopoietic stem cell? So as Alex mentioned, so I spent really almost 5 to 6 years at Fred Hutchinson Cancer Center. And while I was working with Dr. Grudin, and I also working with late Dr. John Hansen and Dr. Paul Martin, I was so curious about bone marrow transplant that just for me to learn. Then I realized that, as you all know, the bone marrow transplant is the treatment for the treatment. If you get a leukemia, high dose of chemotherapy, wipe out your immune system. To rebuild the immune system, you need some cells from a donor. Most of the time, donor cells will attack the patient's body. That is called graft versus host disease. And the real question is, you don't need a donor. Can you use your own cells to reprogram and put it back? This is a little bit a complicated problem, right? I'm not going to say that disease can be solved, but it is possible, right? And this slide, in fact, Dr. Paul Martin helped me to construct this slide like a few years ago. Right, so question is human fibroblast, can you create a HSC cell? In my mind, everything is possible, really, right? Everything is possible. But the thing is, we need to understand inside the nucleus, it's like operating, it's like a computer. We need to understand more detail how the things are organized. So my ultimate thing, I will just put it the slide in the beginning, building a tissue. This is called Turing system. And Turing system is named after Alan Turing. And as you know, Alan Turing is famous for, in computer science, Turing machine. But the last paper Turing wrote is an amazing paper called Chemical Basis of Morphogenesis. So that's why I will explain this slide in my last few slides, but just want to give you, these are like my dreams I want to do. Right, so let's go into this. What is the normal development? Normal development is so beautiful, right? Means that as you saw in my second slide, so I am from Sri Lanka and my wife is from Seattle. And I always wonder, my daughter, that she came from mixing 2 genomes, right? My genome and Lindsay's genome, but it's a beautiful process. Embryonic stem cell give rise to all the possible cells in the body. In fact, before Hal Weintraub and Shinya Yamanaka, people thought it's a unidirectional process. And that was changed. In my mind, that was changed in 1989 when Brilliant Hal Weintraub showed to the world that you can change a skin cell to a myotube directly. That is the beginning of thinking about cellular reprogramming. But unfortunately, he passed away in 1995. But then Yamanaka, Shinya Yamanaka came to this picture and showed to the world in 2006, he can change a skin cell to an embryonic stem-like cell with 4 factors. Right? And I got so obsessed with this for so many years now. When I first knew this, when I was with Dr. Grudin, that means you can change one cell to another. So frankly, I read Yamanaka's papers so many times last few years to understand, go to his head. But his thinking was so simple that he looked at in roughly speaking, if I may put my hat as a mathematician, that he looked at the embryonic stem cell, just what genes express, and the skin cell, what genes express. Then he just subtract. And then he found roughly 28 transcription factors make that difference. Then what he did, he spent like 6 years trying to find the best set. If you want to introduce exogenously to fibroblasts, then what are the best combination that will eventually look like an embryonic stem-like cell? Then he coined the term induced pluripotent stem cell. That's so remarkable. And he got the Nobel Prize within few years. As you know, in electrical engineering, that one of the main topic in engineering is something called controllability, control theory, control. That means give me a target, you have initial conditions, how you can steer the initial condition to the target. In fact, I remember I had so many discussions with Dr. Grudin, like 2011, 2012, but that's okay. Like I never knew that much. And then I got so obsessed when I moved to Michigan. The first thing I really thought I really want to understand the dynamics of a skin cell, how the skin cell go through the cell cycle. So we wrote a paper in 2015, human 4D nucleome. That means so we look at a time series, RNA-seq and high-C, there's a technique called chromosome conformation capture to study how the fibroblasts proliferate. Then that experiments guide us to construct some sort of a universal algorithm in 2017 called this paper, Algorithm for Cellular Reprogramming. I will tell you a little bit about this. I really want to tell you this, right? I really think that, always I tend to believe that beautiful biology, right? And we have amazing technology. I think we need little bit mathematics. We need to come to mathematics, but appreciating biology, data-guided way to get more detailed picture what is happening. So that is what we did. That is what we did in 2017. But again, go back again, 1 minute. So classical theory of Yamanaka is going back a mature cell, reprogrammed to stem cell, going back, right? But most intelligent way in my mind was Dr. Weintraub. He did directly, mature cell to another mature cell, directly, right? And I am in that lineage. And I want to think that way, direct cellular reprogramming. Right? That's because Dr. Grudin and Dr. Weintraub done so much things together. And by the way, Alex and everybody, I have all the old papers of Weintraub. I constantly read to go to his head how he's thinking about biology. Anyway, so now I will introduce to you this idea of data-guided control. We dub this term data-guided control. The basic idea again, if I put a small cartoon that you can get any cell from anywhere, you can put it back to somewhere else, right? That's the basic idea, right? So here, please bear with me. Please bear with me. I really want to tell you this, okay? So the idea of engineering is and math too, that controllability. That means if you know the initial configuration, you have some sort of a desired configuration, how you can move that initial configuration to desired configuration? What is the minimal input required? right? This is very simple in some sense. You can write as like this, dx over dt equals a of t x of t plus b u t, right? This is very, very, in very simple. But you can think the same setup in the context of cell biology, right? We have cells, we have transcription factors, we have data. Those are like RNA-seq, Hi-seq. We can see now the picture, what is happening inside the nucleus, right? And transcription factors are subset of proteins that can do something. In fact, I like to use the word transcription factor guided control. Transcription factors are amazingly powerful, right? And so this was the team we built this setup. And Scott Ronquist was a chemical engineer, and now he's at MathWorks. He got his PhD, and Jeff was a postdoc with me from mathematics. So this amazing group of us, we spent a lot of time to build this setup. But if you have a time, please check this paper out, Algorithm for Cellular Reprogramming. But the basic thrust is here. We use data. As I said, that 4D nucleome is a very big project in my understanding at NIH, that to study genome as a dynamical system. By the way, I don't have any, I'm not a part of that. As you saw that all my funding is coming from Air Force and DARPA. So I'm not a part of that, but I was somewhat responsible from the beginning to think about the 4DN. So that the idea is to looking at the genome as how I can see is a dynamical system that is changing over time, right? So that's exactly what we did in, as I said, the skin cell. That is the initial conditions for Yamanaka as well as Hall-Weintraub, initial conditions. But I put some slides a little bit randomly. I'm very, very sorry. Okay, so because this technology, I want you to, I don't know how many of you familiar with this beautiful technique called chromosome conformation capture. So that is a technique to map out inside the nucleus, like a network, right? Everybody knows RNA-seq, right? The Hi-C is, at the end of the day, will tell you pairwise interactions, pairwise, like a matrix. Or you can think as a network inside the nucleus, right? And so we use this kind of data. In my lab, we generated Hi-C and RNA-seq time series. So that's this picture. And then we use this data, and there was amazing observations we saw. This was in 2015. That means that genes have amazingly coordinated over time, some genes. That means they're expression coordinated. So this idea is very, very related to this concept of transcription factories. Transcription factories. This was coined by Peter Cook at Oxford University. Peter Cook. So we saw this kind of phenomenon that over time, certain genes in certain blocks, they coordinately activated. So we use this understanding. At the end, we create a simple algorithm. I'm not going to this one, okay? So we use all this data we generated, Hi-C, RNA-seq, as well as the things we know, transcription factors and some math. And then we were able to, so again, in this whole setup, we need to think very simply. We need to think very, very simply. If I may draw something, let's say I will draw. So how I think this is, let's say, please disturb me, please. So let's say fibroblasts, right? I want to know something about fibroblasts. Say this is gene expression, say RNA-seq. And also we know something about how these fibroblasts inside the nucleus, how this is organized. This is from Hi-C technique, Hi-C. I hope you all familiar with this technique, chromosome conformation capture. So we have a good understanding of the initial conditions. And then the real thing is, right? Real thing is, give me any cell type, some interest. So you have this is my target. Maybe Alex needs a certain cell type X, but I need to know something about cell type X. You need to tell me what is the signature. Do you know anything about cell type X? Then we can able to reverse engineer the set of transcription factors, the minimum transcription factors. That's the key. The controllability means minimum time, minimal inputs. So this was again 2017, what we did. The idea was to give exogenously a transcription factor, exogenously, right?
Indika, when you say that we need to know something about the cell, do you mean the cell type's gene expression profile?
Perfectly, exactly, Alex. That means you know something about the targeted cell type. Gene expression is fine. And then in my mind, in how our thinking was that initial conditions is the key. I need to understand good about my initial conditions, right? So the experiment if I recap what we did in 2015, what we did in as you know, the cell cycle again, G1, S, G2 and M. What we did, we create a time series. At 1.5 cell cycles, fibroblasts. Every 8 hours, we collect samples, genome-wide, RNA-seq and Hi-seq. From that, we had a good idea how normal proliferation happened. So that was our data we used to generate that algorithm. So, please, Alex, thank you for asking that. And so we were able to, again, that paper we laid out nicely, and we were able to reverse engineer the things we know easily, like Yamanaka factors. And I always love Dr. Weintraub's MyoD. In fact, I revisited Dr. Weintraub's experiment in 2017, and to see what is the time. I always like time series, like how things evolve, right? So just give you an overview here. This is, in my mind, we use the beautiful biology, how the organization principles inside the nucleus, data, and some math, clean math, simple math. And this was 2017. Now we have a different extension of this. So this is my group, what we are working on. I just want to share with you. It's so nice to have smart students around. I was so fortunate here to have so brilliant students. So these are my team here. Again, I want to introduce to you. So, and Walter, Lindsay, and Anthony. Stephen just finished his PhD. He went to MathWorks. And Drew is here. Christopher York, Sam Dilworth, and Can went to Harvard for a postdoc, and Gabriela Doston. So this is the team. And now I will tell you what we are doing. But this is the extension of reprogramming. Right? And like a lot of people think most of the time I am in the computational biology and math, and I don't do experiments. I really do experiments, really, in the lab. And my philosophy is that I always like to get foundations from mathematics. And then I generate my own data. And then I have a good idea what I am doing, right? So the whole philosophy is we have some understanding from, as I said, the data guided theories, but we need to refine this algorithm constantly, learning from data. Right? You have to go back and forth, biology, computational toolbox. That's how you minimize these to get the best set. Right? More and more we can get refined data, we can predict these recipes very, very easily. That's how we believe in. So that's a little bit, I want to tell you some things because of the company I started, and there's a part of that. But again, for me, I just want to be clear, the company was to humanize my science, not to get profit for me. I love to humanize my science, right? That's why all I did that to start this I reprogram. And this is the latest. I think this maybe already people know this. This is the basic setup of transcription factor guided controllability. Activation, this is classic, cell type 1 to cell type 2, like Yamanaka. And also 2020, I was working with a friend of mine at NCI called Thomas Reed. What we did, we sensitized a cancer cell suppressing a transcription factor called TCF47L2. So that gave me the idea, maybe we need to do some sort of hybrid way, not just exogenously giving a transcription factor, we need to do both. That means activate and suppress together. So that means, as an example, first thing is that you need to loosen up the system first and then give a control to give the direction to your target. But the most important thing is, as you know, that these are dynamics, right? We need to do this introduction or suppression in a time series manner, time dependent manner. Because certain stages, the genome is more receptive to do something. So that's the one. More and more even the COVID time, I was spending a lot of time on the last setup, how to find more refined recipes using hybrid. I really want you to think this too. And this is so beautiful. That's what I said, that there are a lot of technology, right? We can get RNAi, modified mRNA, all these are there now, right? We need to use most intelligently how to do this. And so as you know that when we are in universities that we can get nice equipments, there's a program called DURIP, Defense University Research Instrumentation Program. So I got a lot of gadgets into my lab to do this. I just want to show you that. One thing, I'm constantly learning about the time, how the cells go through time. This is the live cell imaging system. And then also I'm using a lot of sequencing, very simple sequencing, right? And NanoPo. I'm using NanoPo lately. So all these things we do, we guided by data. But as you know, data is always incomplete, right? And we need to use some imagination here to how to put this together. So because of this, we were able to, I was so obsessed with, my funding is coming from Air Force, one part. They are very much interested in wound healing. So one day I realized maybe we can use the same ideas like reprogramming. Can we accelerate the wound healing process using transcription factors? But wound healing is a very intricate process, as you know better than me. That's when you have a wound, then there are two things happening, migration and proliferation. At the end, wound will heal. But we are using a very simple prototype, in vitro prototype for wound healing assay. And to see, can we accelerate this process? Indeed, we were able to accelerate this process recently using hybrid control, hybrid method. What I said that this is again preliminary results, that taking out a transcription factor and giving a transcription factor together. This is our findings recently, that this is a normal recovery. Again, we are far from in vivo setting. This is simple in the lab, in vitro. This is the normal setting, normal recovery. This is with interventions. So here we use to suppress and activate. We suppress PRAX1 and TFAP2C. These are some candidates, and we had the system MyoD. I always like MyoD. This is a very surprising finding for us. But again, this is an idea using hybrid techniques, hybrid. But again, you might ask, is this really reprogramming? Indeed, this is reprogramming. Here, the idea is that how I have been thinking in this setup is that you have a stable state that, like say normal, say fibroblast. This is simple scratch assay. We are going further, introducing keratinocytes, go more. But the simple thing is you have a stable fibroblast, you make a disturbance. You make a disturbance, right? You disturb the system. And then at the end, there is a process of wound healing, right? This is the healing phase. Healing. And go back to, ideal case, they go back to stable, right? That's the idea. It's amazingly this during healing, there are two processes are happening, right? Migration and proliferation, right? They are amazingly together, this healing happen. What we doing is we intervene into this process to accelerate this process. So that's what I just show you, that our simple findings here, right? And using, this is far from to publish, or still we, and I'm happy to talk someday with anybody with this system. And I'm so fascinated with that. So we are now automating this system. We are automating this system now. Oh, sorry,
Indika, what's the rough improvement there? Is that like a 50% time? Like, I'm sorry, I just didn't.
Oh, so the fraction of wound recovered. So purely this is from Alex, imaging. We can look at the imaging and how long it's recovered. We can measure the number of cells and the area.
Yeah,
So,
I get that. I was just asking what's the, it looks like a pretty good effect size.
yes.
I was just,
Almost like if you see the more than double, right? We can completely accelerate the process.
yeah, that's what I was wondering. It looked pretty significant. So yeah, more.
Yes.
That was the take-home message there. Great, thank you, Indika.
Yeah, Alex. And as I said, so this is an application of reprogramming, but more and more we need to understand what is happening inside the nucleus, right? That is the operating system, right? So, Alex, I have now another half an hour, right? Half an hour or 20 minutes.
20 yeah, minutes. I would give about 20 minutes just to give people.
Okay. So let's recap. So what I showed you is something we done about reprogramming and what we are doing now to accelerate this process. But we also want to really understand what is happening inside. In the previous algorithm, I show you that data, we use this idea of chromosome conformation capture, right? But there's another extension to that now, that genome is not just pairwise. There's a higher order interactions in the genome, right? And what we did the last 2-3 years, I always like to build mathematical framework first and then try to do the experiment after that. So we did some work on some concept called hypergraphs. So the hypergraph means that like this, let's give an introduction. Hypergraph. Normal graph is just pairwise, like graph like this, right? Bob, Amy, Carl, Ed, Dan, just simple pairwise. It is like Hi-C, right? Hi-C, pairwise, right? By the way, now there are extensions too, using other, not alumina, using other techniques, you can get maybe multi-way interactions. But usual Hi-C is just pairwise interactions in the genome, exactly like this right figure, right? The hypergraph means the next extension. If you see this picture, this is a simple example that Carl, Ed, and Dan, this share one edge. These are called usually in network science, we call this Carl, Ed, and Dan is like a node. And the red part is one edge. So here you have a one edge between Carl, Ed, and Dan. So that will tell more information, right? That will give us you can see the relationships unambiguously, right? This is the idea of hypergraphs, right? So again, at the end of the day, Hi-C is a matrix. It's called a matrix, right? It's a linear algebra. And the next extension is something called tensor. That means you can go beyond two, right? That's you required some hypergraph ideas. And I'm not going to tell you about hypergraphs. So there are some papers we written purely mathematical ideas. Okay, we build the framework, hypergraph, controllability of hypergraphs and hypergraph comparisons. And that allows us to this new technology, this called POSI. POSI will tell us, because of this is called long-range interactions between genomic loci. You can extract, really, I will go to the next slide directly. So this is the flow. Because of this technology POSI, you can see more than 2 higher order interactions. Remember I said that at the beginning, this notion of transcription factories, right? The idea is that many 3-4 loci come together in space and use the same transcription machinery. So this technique is a beautiful technique to see that. Again, this have a lot of problems, this technique, but a lot of problems. This is 4C, lot of problems, but again, math, little bit math, we can get the extract some features from this data. That's what we did. That's why math is again very powerful in some sense, right? Extract patterns. So here the idea again, if I may spend like few minutes here, I really like you to think about, maybe our data is a lot of noise, but the idea is that to extract higher order interactions in the genome, higher order, that will allow us to see much better what is inside, what is that compute, how that the architecture inside the nucleus, right? So we can see this lot of patterns. These are like motif structures inside the nucleus, motif structures. And this is that if I may explain this tiny bit, the idea is that here you can have pairwise, you can have 3 loci, you can ligate, 4. Because of long reads, we can recover that. The natural way to represent this is another way to represent this data. This is called incidence matrix. Like this is reads purely from data. Right? The these 4 loci, you get a 1. All these 4 loci, that's a read. So that means you can think this is like there are 4 nodes and a 1 edge. Right? Then you can see that how this organization principles. And this allow us to go a little bit beyond now. As I said, I think Alex asked me, we need to get a good understanding of the targeted cell type, what target I want and what is my initial conditions, right? I think this technique will allow us to see a little bit more refined way that to get a signature about the architecture of the genome architecture, not transcription, genome architecture, how genome is assembled inside the nucleus. And then this is also allow us to fusing data. We were able to extract transcriptional clusters. Like I said, transcription factories. We use the word transcriptional clusters inside the nucleus. So this is a more refined way to see what is happening inside. What is the picture? You have all these slides I will share, but right, you have all these slides. And this is again very simplified way of looking at different type of data and put it together and see how inside the nucleus, how that is organized, how that transcribed. Are there any principles? That's the whole idea here. If you know more and more, we can reprogram. That's how I think. More and more we know, efficiently we can reprogram the cell. And please disturb me. It's not make sense. So we were able to extract beautiful patterns inside the nucleus because of this technique. This paper is in the archive now. And
so Indika, if I understand you correctly, what you're able to do now is you're starting to understand the spatial organization of chromosomes and then the rules for transcription factor binding.
Exactly.
3-dimensional space by instead of just being able to see one or like two genes interacting.
Exactly.
Partners at once.
Yes.
And then cool. And then you'll be able to visualize this at some point.
Yes, exactly. That's why imaging, we can verify that too, Alex.
Wonderful. I just want to make sure we all understand because this is very, very cool and very high level stuff.
I have a question.
Yes.
So, are you doing this as, like you said, for your earlier stuff, you did in a time series manner where you looked at different stages of cell?
So, Mahdi, for now, Madil, thank you, Madil. So here, so far, because to develop this thing, we only did Madil population level, synchronized population. Now we are in the phase, we are now going to do everybody like single cell, right? We are doing now single cell POSI. And this is a joint project with Peter Fresh at University of Florida State. And not a time series yet, not yet. This is an aggregation of many cells. So that's what I said, some sort of biological interpretation is not that crystal clean. but the conceptual framework is good to looking at this in this way. So in my mind, this paper, we lay down the conceptual framework, some simple mathematics and data, no time series yet. And anybody interested in this technique, I'm happy to help with any way I can. We know this technique, population level POSI, and many people doing this too, I think now. And we can interpret this data very simply using hypergraphs. And I have a few minutes, but I hope that my goal was not to tell exactly, to give you like how we can put engineering, beautiful biology and math together. I think three required to get magic. But I want to share with you something, go beyond that, right? Go back to Clark again. But as I said, Turing, right? Turing system is really indomitable. I'm working on a paper writing this. Turing system is indomitable, right? How he conceptualized the how morphogenesis conceptualized. It's a beautiful paper. I highly recommend. You might have seen this paper, 1952, right? How he thought about morphogenesis. It is some aspect of reprogramming. The reprogramming is engineered the system, right? And here, when he construct this idea, he never knew the genome. He thought cells has chemicals, right? No genome. And as you know, Dr. Smale, who I work with, Dr. Smale touched this problem too, before even I was born, 1976. Right? He touched this problem. He go beyond Turing. No genome. But I was so fortunate to work with Dr. Smale in 2017. We revisit that with genome. So this is again a mathematical paper. We build the mathematical theory, how to combine genome with cell dynamics, right? How to think about a tissue. We need to not just reprogramming a cell, we need to understand the tissue. So this is again, this paper is in a little bit of mathematical paper, but we lay down the Turing system, bringing the genome. But that's why I really believe people have been doing this. We can someday print a tissue just from your skin cell. People have done recently using embryonic stem cell, iPSC. But as I said, I'm coming from Weintraub's lineage. I want to do just from skin cell directly, construct all the cells required for a tissue. And using, there's another technique I didn't talk in my lab. We are also into spatial transcriptomics. That is a technique that will allow us to see the patterning of the tissue, which genes expressed in a tissue. So using these ideas, spatial transcriptomics and reprogramming, and some aspect of extracellular metrics. This is like biomedical engineering, right? So you can create these biological scaffolds. And I really think within 5-6 years from now, we can print a 3D print tissue from your skin cell from somewhere. So this is like my dream. I want to tell you that this is my dream. Really hold these things more and more. I want in my lab to understand the operating system inside the nucleus, and more detailed understanding about data and math and put it together, right? And go back to that. And this is a paper, again, you might not even find this kind of papers. And there's another long paper I wrote about thinking about the genome mathematically, called Mathematics of the Genome. And this was guiding to me a lot lately to think about these things, the data, and how we can conceptualize data a little bit elegantly. And I think I will stop here. And I hope, my sincere hope is that to give you an idea that I really think that we need to put these three things together is most fundamental way. Beautiful biology, beautiful mathematics and engineering, right? And I am experienced personally. In fact, I was telling to Alex that I have a course in University of Michigan last 6 years. I started with 12 students called Mathematics of Biological Networks. In this semester, I have 76 graduate students and 46 from biomedical engineering. Right? And that's the thing, I'm so excited for future to put these three things together to humanize our science, right? Really want to humanize our science. And Alex and all, thank you so much for listening to me. I really like chalkboard to write. So my slides, I'm so sorry if the slides are not coherent. And again, thank you so much for your time to everybody listening to me.
Indika,
Thank you, Indika.
for everyone, that was fantastic. It was really exciting and a really, really innovative approach that I'm really excited that you're able to share all of your work with the department. And yeah, I'm sure we're going to have a lot of questions because it's really innovative. And I immediately see applications for aging and have a lot to talk to you about normal tissue heterogeneity and how do you fix things and blah, blah, blah. And a lot about that.
Yeah, Alex.
There's modeling to be done there and advanced math to think about and probability theories and, thinking about adaptation to stress as a function of blah, blah, blah. So anyway, let Jason, did you have a question?
I've got about 5 questions, but I'll start with a simple one. So and this sort of relates to the other question that got asked earlier, but just conceptually, could you build the hypergraphs from purely time series data? So even if you weren't looking at spatial correlations, could you do it with temporal correlations?
Absolutely,
and would that actually,
Jason. Absolutely,
okay.
Jason. Absolutely.
Because I would think that would allow you to use, sort of much simpler technologies and actually basically derive the same relationships, right?
Absolutely. Absolutely, Jason. Absolutely. Let's do something together.
Okay. I'll see if anybody else has questions before I ask the rest of mine.
Yeah, anyone with a question, Indika won't bite. He's very friendly, and I think you'll find him more than welcome.
Please, yeah, and I'm so sorry. I always like to think Alex and Jason, because as you know, like DARPA, they want to think a little bit ahead. So my mind is now reprogrammed the way like that. I like to think anything is possible if you put your head right. So
let me ask another one of my questions. So and you sort of touched on this a little, but like so wound healing is obviously a great application of this technology. But yes, I would think that an even more transformative approach for deriving these sorts of interactions in minimal sets would be dealing with cancer treatment. So that when you look in the course of tumor development, can you identify the minimal set of targets to basically to inactivate a cancer? Have you guys started doing anything with that?
Jason, thinking about this, I'm very closely worked with a person called Max Visha here in the Cancer Center. And Max and I are now lately, we are constructing some POSI data for cancer stem cells. There's an idea called cancer stem cells, right? So I really believe, I think that in that case, you have there's a little bit, Jason, if I may say that, all the things what we do, we assume genome is fixed. There's no aberrations in the genome.
Right, yeah, right?
Sure. When we talk about cancer, we need to understand the architecture. But again, like Hi-C and POSI is in some sense the high resolution karyotyping. We can understand that, right? If you know that, absolutely, that your target is like, can we put them to senescence? That is your one target,
sure,
right? I always ask from people. Exactly, Jason. I always ask from, do we know the signature? Can you give me a signature? That's a minimal signature, that cell will die, right? So that is the problem, like Thomas Reed and I started to do in this 2020 to sensitize the cancer cell, taking out TCF47L2 transcription factor. I really think someday we can reprogram a cancer cell. I really believe that, and very like persistence, and somebody will do very soon. There should be a way to reprogram a cancer cell.
That's
if I want, Jason, I love to be involved with that, a project on that. Right? Because finite number of transcription factors in the genome, right? Very finite.
Yeah, I mean, you do raise the, you're right about the fact that as sort of cancer evolves, you do get these sort of chromosomal aberrations and things. And so like you could imagine that it could escape your ability to sort of look ahead, but
Yeah.
but I think it would still be extremely powerful.
Yeah,
the other question the other question I had was just sort of, and hopefully the answer to this isn't too mathematical, but is there sort of a is there a limit to the sort of higher order interactions that you can see in when you're deriving your hypergraphs? So like, because essentially you're looking at the emergence of sort of complex patterns as things, you know, as more and more nodes in the network are sort of correlated.
Yeah.
Is like, it's some like, can you sort of zoom out and see like even larger scale?
Yeah, Jason,
like,
so this is the technology we are using, nanopore long reads. I think now, like, what is the other technique in not the nanopore, what is that? The PacBio, right? You have more long reads, right? It's depending on the integrity of like the read length too, when we are constructing from data, hyper edges.
right. So it's entirely that. So right now the limitation is based on the actual the nanopore technology. So
Yes, yes.
So do you have a feeling for the like absolute number of?
So we have seen
like
this. Yeah, we have seen up to like 15 node hyper edges.
Okay.
15, right?
Okay.
And this has, that's why we need to do more and more. Are they really noise? And we need to verify, as Alex said, from imaging. Right? There's this technology, right? You can create some oligoprobes and see whether really they are close in space.
Yeah.
Right? But for me, again, I love hypergraphs, by the way, Jason. I love now more and more hypergraphs. And you can check it out, a paper we wrote that how called controllability of hypergraphs. But this is a bit hard though, Alex. By the way, I want to tell you these hypergraphs that, and 2020, a good colleague of mine in University of Michigan, Roger Cohn, and he's imaging the brain, is mice brain, and they were recording like 20 neurons over time. And then he got a time series imaging. Then one day he told me somehow he knew that I'm teaching networks, and we talked, and he said, can you help to understand this? What is happening? What their experiment was: fasting, give food to animals, and not and food, fasting, and food again. And to understand this simple network, what is happening. Really, so I thought that this is a really good example of a hypergraph. That means, by the way, Jason, from correlations to you can construct, you can infer hypergraphs too, called multi-correlations. You can infer.
That's what I was basically getting at.
Exactly, you can infer that. But POSI is the technique, Jason, that will give you that truth.
Okay.
Right? Sorry, Jason.
That's okay.
But the multi-correlation, so we submit that paper, they got rejected. That is, they are too much to use hypergraphs. Then we went with just graphs, right? So that's I think the hard part sometimes when you use little bit, you can publish in math journals, but little bit hard time to publish in a real biological journal, Jason.
That's too bad.
Yeah, but that is you are absolutely right. You can infer multi-correlations from correlations, Jason. In fact, that is a technique I'm working on with a student of mine, and that we know the truth. Let's say POSI and high C. How much we can get from high C to POSI, like what is the passage?
Right. That's exactly where I was getting.
Yeah.
That's very exciting.
Yeah, it's so exciting, Jason. That's why I don't know, Alex. I feel now I am in still in high school, just starting. Seriously, there are so many things I realize don't know, and so many amazing problems to tackle, right?
Yeah, that's just how it keeps going. The more you know, the more you don't know.
Oh, I realize I don't know so much.
Well, I mean, it's a big wide world. It's really cool, Indika, that you're able to take the perspective you are, and I think right now is a really great time for getting it, 4D nuclear architecture with all the advances in imaging technology. We're excited about it in our lab because we're interested in heterogeneity with age and non-Mendelian patterns of it inherent.
Okay.
Why do genetically identical animals do different things? Like what's up with that? How much of it's epigenetic, and then how much of the epigenetics has to do with nuclear architecture, right? And
Alex, absolutely,
that's
Alex.
Probabilistic versus programmed, and how do you influence that?
Yeah.
They're influenced spatially versus non-spatially, because it seems like you can infer a lot about gene expression just based on where some genes are located, but then other papers have now come out showing that there's genes expressed in the middle of heterochromatin.
That's right. Yeah, absolutely, really, Alex. So I didn't talk about that, as I think you said you saw that our paper that we were able to take out the mom and dad and 3 different time points, the high C and RNA-seq. It was really nice observations, but now someday we can do with POSI. You can study more, right? And,
it's really, really interesting work. I think there's a lot of, like you said, you've already begun to humanize it well. I really liked the figure with the fibroblast and just seeing the time series and that you're able to double this, the model of wound healing is still, I'm sure the defense agency people were excited about that.
That one, yes, Alex. So we are now going a little bit more than that to create so I'm in the biomedical engineering. There are a lot of really amazing biomedical engineering students that we can create go not just the one layer, we are going for keratinocytes. Then you have 2 layers.
Oh, fantastic. Wow. So you're actually getting to multi-layered tissues and real tissue. That's really important. I want to make sure before, I'm having a great time with you, Indika. I'm definitely going to have a lab meeting with you in some point in the future, if you're willing, with Jason.
Absolutely.
Because Jason and I have some interesting imaging stuff that might be, it might be to think about networks inside of animals, and then actually, the animals themselves is sort of a little network. But there's this thing where Jason and I have seen animals sort of dying, like there are patterns where their cells fade out, right? The animals sort of fade to black. And if there's anything to be learned from that pattern of death,
yes,
pathology to be gained there, maybe you would be sort of the person to think about that with. Math.
Oh, Jason, and we can absolutely. I love to learn from Jason.
Yeah. Well, Madil, anyone else? Does anyone else have any questions for Indika? I want to make sure we're not hogging all the Indika time. Anybody else?
I had one question. So just you're saying high C or POSI or in combination, how good is the prediction of gene expression?
Oh, so as you know, the original even high C paper in 2009, that what they really found was that what's the conclusion of high C 2009? That means the euchromatin is more preference to nearby to euchromatin. Heterochromatin is more preference to hang out with heterochromatin, not vice versa, right? That was the basic finding in 2009. And also they found that the genes in the euchromatin region, they express, right, naturally, it's the gene in euchromatin region. But there is if that is the case, there is some correlation with high C signal. So Madi, could you please tell me your name again?
It's Adil, actually.
Adi. So
yeah.
So so how I think is that because of this idea of transcription factories, right? There should be a relationship between genome structure and gene expression. But I don't really know that, as Alex pointed out, like a lot of cells we are working on, like fibroblasts, they are not fully differentiated. Naturally, not fully differentiated cells has some uncertainty, right? They are not fully different. I tend to think differentiated cell has like a universal patterning in even 3 space, where they transcribe. That has to be hardwired in some sense. But answer to your question, I think there should be a relationship. We don't know yet that really how they couple. But in my favor is, I always tends to think geometry precedes biochemistry. That what I mean by the geometry is how the genome is organized in 3 space. Right? That's how I think. Just learning from Dr. Grudin, really. What is your background?
Go ahead.
I'm a first year PhD student in Department of Pathology.
Okay. Hey, please connect with me anytime.